Good afternoon, everyone. I'm Sonal Katyal, the Communications Officer of NCBS. It's the Brain Awareness Week, and we have brought you to another lab where they study a lot about, computations and sequences in the brain. But what are these about, and whose lab are we talking about? So this lab is headed by one of our faculty members, Professor Bhalla, and today we have two members of his lab, who will be telling us how they decode the computations going inside here. So let's meet Anil. Hi Anil, tell us about yourself.
Happy Holi, and
Happy Holi.
Anil from Professor Upinder Lab. I'm a graduate student here, and I'm basically working on how the brain is so temporally precise. Okay, so
what do you mean by temporally precise?
Yeah, let me explain this slightly in detail. Okay, so you have all played probably cricket and all, right? And while learning to bat, you have to predict beforehand when the ball will arrive and when you have to swing your bat.
Right.
But if the signal that's telling you the ball has arrived is not exactly timed, if it is coming 1 second later, or sometimes it's coming 1 second later, sometimes it's coming 0.5 second later, sometimes it's coming 1.5. So it will be very difficult for you to learn. Right. So that's what I mean by temporally precise. The brain is able to transmit the signal in a very at a very particular time,
precise and like very okay. So even like a few, like half a second delay can actually cause a lot of problem, right?
So what we do is
what is this?
So these are neurons in the brain. And what we do is we record, not I particularly, but our lab records from these neurons. They inject some current, and then you will see something like this.
Okay. So we are giving current to these nerve cells, right?
Yeah. Okay. So what basically it is so like we all have electrical wires in our houses, right? These neurons are also like that. They transmit electricity, but their magnitude is very low, so you won't feel it. But that's how they just like electrical wires in our houses. The neurons also transmit electrical signals.
Okay.
Yeah. So
lots of electric stuff happening inside our body as well.
Yes.
Oh wow.
Yeah. So what it does is, when there is a signal, say you see the ball coming while you are playing cricket, the sensory neurons in your eyes pick up those signals and then transmit these as voltage. Membrane an increase in voltage. So if you see here, there is a baseline voltage, and then the voltage suddenly increases because there is a signal here.
Okay.
And then it transmits that signal to more neurons further downstream, and then more neurons, and then collectively all the neurons combined make the decision whether to take a left shot or a right shot while you are batting.
Okay.
So what I am trying to do is,
so much is going on when we are playing or thinking or moving, and we have no idea.
A lot of stuff is going on. Yeah.
So everyone can see these signals. It's not your electricity thing happening at home. It is something happening inside our brains.
Yeah. So what I'm trying to do is, I'm trying to understand why this temporal precision. So I'll also show one thing. So this is when we inject current into
like
the
holy.
Yeah.
So many colors.
Yeah. So when we inject current into the brain, you will see that. So every time the spikes are occurring at the same time. So these are called spikes or action potential. These are occurring at very similar around the same times. So like here, or in this way in here. But sometimes what happens is, in diseases like Fragile X syndrome, which is the autism spectrum disorder, the precision is gone. So for example, if you see here,
Oh, now
spikes are,
the spacing has
yeah,
changed, no? This
yes,
one is like broader. This is
yes.
okay.
That's one of the
Can we see the before? Like this
yeah.
is so. This is normal functioning,
This is normal
and
functioning.
the
And
other one is with disorders.
yes,
Okay,
yes.
that's quite interesting.
So I'm trying to understand why this is happening in Fragile X syndrome. I'm trying to understand what are the role of certain proteins that are there in the neurons that
Right.
help with regulation of, I mean, with that help in giving precisely timed action potentials. And I'm building basically computer models of these neurons. So this here is the experimental data. This here is the model that I built, which match so that I can predict what all proteins or ion channels are contributing to this uh, imprecision.
Okay,
So
great.
that's my work. Then in our lab, various other computational projects are going on. So we see computations happening at various levels. For example, in networks. So I saw these neurons, right? These are connected to each other, and they form a network. And uh, what are the computations happening there? Then you go below that to neurons, one single neuron. That's where I am working on. Then even you go below that, and you'll see there are dendrites in the neuron where
Dendrites are
the small, yeah, you see. Okay, so these, okay, it's not visible here, but these small, uh, very tiny.
so the
Yeah.
ones going from the axon.
Yes.
These
Yes.
little ones. Okay.
Yes, these are dendrites. So there are various computations happening at dendritic levels. Then within the dendrites also, the molecules that are present in the dendrite, they interact with each other to do various other computations. So all of this work is going on.
Oh, thank you so much,
Yeah.
Anil. That's really fascinating. We never expect, so much is going
Yeah.
inside our brain. And those who are watching us live, if you have any questions, please do write in the comments and we'll try to answer in the live itself. So now we have, thank you, Anil. We have another lab member, Anzal. So, hi, Anzal. Tell us
Hello,
about yourself.
I'm Anzal. I'm from UP's lab. I'm a fourth-year graduate student.
Okay.
So, as Anil had told that there are several computational projects going on in the lab, there are some experimental projects going on, and the scale of these experimental projects starts from behavior of the animal and understand how information is stored in our brain
Okay.
and in a network fashion. For
Okay.
example, uh, there are a lot of things that could happen inside the brain, right? When you go to a restaurant, you know that it's a restaurant. When you come home, then you know that you are at home. So how do you understand that you are in different places? So this can be achieved by a lot of neuron working together
Okay.
and a lot of information coming to different sensory organs that you have.
Okay.
For example, you listen, you hear, you smell, all those things happen, right? Now, all of these information has to go through different parts of the brain and then converge to certain other parts of the brain. So, sensory organs sense this information and send it to brain.
Okay.
From the eyes, it goes to brain. Inside the brain, different parts are there, and some parts send that information to another part of the brain.
Okay.
And such one part is called hippocampus, where you have a convergence of these sensory information.
Okay.
And it has been shown in animals that they remember certain places that they are interested in. For example, if they find food, they will go back to that area,
Yeah.
right? So,
Or like a secure, safe place.
yes.
Yeah.
So these things are either called place cells, grid cells, and so different kind of cells which respond to a particular kind of context they are in. Now
Okay.
the question is, if a cell is getting a lot of information like that in a sort of a pattern of inputs, how will the cell know that the pattern of input that it got at that moment is relevant or not?
Okay.
Right.
So how is it like sifting through all the information it is
Yes.
getting?
And now cell will have to decide whether should I rely this information further or I just stop this information flow at this point.
Okay.
So in order to do that in lab, as I started, there are behavior projects going on, and in my project I look at it in a single cell level.
Single cell level.
So
Okay.
in order to achieve that,
So I'll just pause you. We have a question from Chandrika. Chandrika, please stay on. We'll get back to you after some experiments that Ansel is showing. We'll go back to Anil and answer your question. So stay tuned and do follow our dive. Please,
Okay.
Ansel, continue.
So now if you are to follow this single cell level of computation,
Okay.
to see how the neuron compute and consolidate this information and decide whether to send that or not,
Mm-hmm.
you need to find small parts of the brain,
Mm-hmm.
and you need to look and listen to those neurons in the brain. For that, you have to take the brain out of the animal,
Mm-hmm.
and like I have done here. So if you
Oh.
look, you can see that this is a mouse brain.
So this is like
Okay.
the full or half?
This is half.
Half of
So if you look,
This is
you
so
can see
tiny.
only half, right? The
Oh,
other half,
wow.
where did it go?
So, in fact, our brain shape is very similar, right?
To this, yeah. It's little different, but it's, many parts are kind of same.
Oh, okay.
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 So now I have dissected out the brain.
Mm-hmm.
Now what I have done is I have taken a part of the brain out of this whole brain and kept it alive. The
Okay.
animal is dead. Animal is no longer with us, but we have that part of the brain kept alive. So
Okay.
that I'll show you.
Okay, we are heading off to Alive Brain, and wow, so we've entered a kind of a very high intensity science going on
So-
here room.
If you look into these,
What is this?
you can see that there are tiny pieces
Right.
that I have kept here, and I'm
What
bubbling a solution.
What
So
are these
this
pieces?
tiny piece is what I told that I have dissected out a part of the brain called hippocampus.
Right.
And if you look at that hippocampus, after putting in under
So
this
these are like
microscope-
pieces of the hippocampus?
yes,
Okay.
these are pieces of the hippocampus.
Slices, right?
They are sliced. They
Okay.
are sliced into tiny pieces
Okay.
where we can actually see if we could keep under the microscope.
Okay.
So once we keep that piece of brain under the microscope,
Mhm.
the piece will look like this.
Okay. So this, when under a microscope.
When under this microscope, it will look like this.
Okay.
Okay.
Wow.
Now, this is little zoomed in, but it's not all the way zoomed in where you can see the neurons.
Okay.
Okay. Now let's zoom in further.
So we are
and
going to one cell.
one
the
cell
one
type
level.
of level.
Okay.
Okay. Now we have this microscope. We zoomed in and we have kept it ready for you guys.
Okay.
So if you look here, you see this line, which is like a tiny teardrop sort of structure,
yeah.
and you see some here. So if I move my microscope here a little bit here and there,
Mm-hmm.
you can see those guys clearly now, right?
Right.
So these things that you see,
So these
these
blocks.
are neurons.
So this is like the nucleus part of it, or
It's the cell body part of the neuron.
Okay.
Okay, so
The popular like
yes,
the circular pentagon kind of thing we see.
kind of thing that you see.
That's what we are
Yeah,
seeing here.
it's looking like a triangle shape mostly because these are specifically called pyramidal neurons, which looks like a pyramidal structure.
Okay.
Okay.
So different neurons in different parts of the brain have different
Yes,
shapes.
different shapes.
Okay. So
And
these are pyramidal neurons.
These are pyramidal neurons.
Found only in the hippocampus. Mostly in the hippocampus. Okay.
There are other parts of the brain also which
Okay.
has. Okay. Now let's say I want to listen to these neurons. What can we listen to?
Okay.
Like Anil told, there are tiny electrical activity that goes on in the neuron.
Right.
And when I told you guys before that if the neuron decide to transmit this information or not, the general way of transmitting an information is to provide a very huge fluctuation in this tiny currents
Okay.
across this neuron,
Right.
this membrane in the neuron.
Yeah.
So what you can do is you can try poking the neuron with a tiny needle, something like this. Okay.
Okay.
So this is a very tiny needle, and what you see here as this triangular shape,
This is the needle.
this is
right?
the needle that we have inserted. So it went from here and it went deep inside,
Okay.
and it got attached to a tiny cell here.
So it's that sharp that it can attach directly
Yes.
to just one cell.
One cell. And if you look, the cell is this big, the cell is this big, and the needle is only this big, tiny one.
Okay.
Okay. So it's very tiny.
Mm-hmm.
Okay. Now you can poke into that cell, and cell and the needle makes a continuous
Circuit. Okay.
Now the question is, how will you listen to this very tiny currents? These are really tiny, that you will never detect in your usual machines.
Okay.
So you need to amplify the signal further.
Right.
So that's why we have all these equipments here.
Okay.
So, and you need to move them in a very precise manner. So
Okay.
that's why we have these dials. These
Okay.
dials help us to move the needle under the microscope. And
So this signal thing is happening there or at this
so
end?
the signal is through the needle. It comes up and it goes through this amplifier. It's a tiny amplifier.
Okay.
And again, that cable goes down and all the way it reaches an instrument kept down.
Okay.
Okay. So that's
And then
an
you
amplifier.
do the adjustments?
Yes. For
And
that,
then-
we have a software ready.
Okay.
Okay. Now, let's see what happens if I try to do an experiment.
Okay.
Okay. So you saw in Anil's model there are spikes
Right.
or tiny electrical pulse changes.
Right.
So if I run this, you can check whether the neuron is firing or not. Yeah.
Oh.
So it's firing like anything. So I'm injecting a tiny current into it and seeing what happens to the neuron.
So it's responding.
It's responding to the current that I'm injecting.
So this reminds me of like Koi Mil Gaya, no? You're sending signals to the other planet
Yes.
and it's responding. So here we are sending it to our something on our own planet, which is the brain cell, and it's responding through these signals.
Yes. So now,
Wow.
so now you know that the neurons can respond to these signals if we send
So each of these spike is a different
different
response.
response.
And does it also mean that so many times the signal was given, or the signal was provided just
Signal
once?
was given. So if you look, there is one line here,
Right.
another line above it, another
Yeah.
line above it. So every line that you see
Is one.
signal that I
Okay.
give. So every step I give, I'm increasing the amount of current
And
I'm
with
giving.
each signal, these are the responses.
Yes.
Wow. Okay.
So beforehand I told that you recollect neurons get a pattern of input, right? Now if I want to give a pattern of input to a neuron, what I can do is I can genetically modify the animal that we are using and make some neurons respond to light.
Okay.
Okay. Now if I use a projector and instead of projecting it to the wall, I converge it and project it under the microscope and put light on places where the neurons are, then those neurons will fire, right?
Right.
Now, this will mimic the input that I told in the brain, where there are multiple neurons sending a patterned input to one neuron that we are listening to right now.
Okay.
So for that, we genetically modified the organism in such a way that some of the neurons which get lights responds and send signal to the neuron that we are listening to.
Okay.
So that's how I'm doing my project here.
Okay.
And if you have any doubts about this,
Yeah,
I'm
please,
happy to address.
if anyone has any questions, please put in the comment section. I do have one.
Yeah.
So what is happening here?
So here, if you think about it, I took the brain out of the animal.
Right.
There is no longer blood to provide nutrients and oxygen,
Okay.
which is very essential for our brain, right?
Right.
So if it's out of the animal, you need to provide nutrients and you need to provide oxygen.
Right.
So how do you do it? The solution that you see is not water.
Okay.
So it has all the nutrients that these neurons need, like glucose, other ions like potassium, calcium, sodium, magnesium, all those ions and all those things to balance this system in such a way that it's assuming it's close to inside the body itself, and in a vat where there is all the required nutrients are there, and the bubbles that you see are the oxygen bubbling.
Okay.
Okay. Now, if you look, I have put a pipe and a bottle here in which there's a solution is there, and I have bubbling that also.
Okay.
And that solution is being pumped and taken through, and I have heated it to the body temperature and then taken under the microscope. So it's going there and it's perfusing basically.
Is there a reason why it needs to be bubbled?
So your dissolved oxygen content in these solutions are limited.
Okay.
And these cells need oxygen. They will consume it every now and then.
Okay.
And the oxygen in the room is not enough to provide oxygen in the solution.
Okay.
So you need to force and pump some oxygen into the solution
Right.
and make sure there is enough ample
Something
amount
that the lungs
blood.
do.
Yeah,
Like
with the
they,
help of
blood, right? So we
Yeah.
are doing that So these you've kept for like when you need more slices to be studied,
Yes.
right? So they are storing the slices here.
Yes.
Okay. So, Rack T is asking what is the name of the instrument used here? Rack, could you specify which instrument you are talking about? Is maybe the microscope?
So we use a microscope which is specifically designed to see the kind of tissue that we are looking at.
This is done in-house or like?
So the microscope is commercially bought,
Okay.
and much of the designs are done in-house.
Okay.
For example, the chamber that you saw here is an in-house design.
Okay.
This whole setup is in-house design,
Okay.
and many of these electronic components, high precision components, are commercially available. Not
Okay.
publicly accessible, but.
And maybe they meant this instrument. Is there a name to this?
There are few sets of instruments here.
Yeah.
So one thing is you need a computer to control all the instruments that you are
Okay.
going to record or try out your experiments with. So that's what you're seeing here.
Okay.
Next, you have a rack full of instruments.
Mm-hmm.
So one instrument is the one which allow us to control the needle here because it's very tiny and the movements are really. It has to be really smooth.
What is this called?
It's called a micro manipulator.
Micro manipulator.
Yeah, which basically means you can move your needle in micrometer precision.
Okay.
Because your cell is like 20 micron size,
Right.
and your needle is like 2-3 micron
Right.
size
Right.
in diameter.
Okay.
Now you have some more equipments at the bottom. These equipments that you see are called amplifiers.
Amplifiers.
Yeah,
Right.
this is two instruments are there. One is amplifier, another is digitizer.
Okay.
Which basically means you get an analog signal from your cell, which is
Okay.
current waveform. You have
Okay.
to convert it into digital waveform.
Okay.
That's what this guy does. And the
Okay.
amplifier is amplifying the signal. Okay.
So I hope, Rack, your question has been answered. But if not, please do let us know more details.
So if you are asking in terms of what the whole setup is called, it's called an electrophysiology rig.
Electrophysiology rig.
Which basically means electrical measurements and physiology is changing, right?
Okay. Thank you so much, Anzal. Could you let's go to Anil back and ask Chandrika's question. So I hope you all have enjoyed, the various instruments and experiments going on in the rig. And now let's find Anil again. Hi, Anil. We have a question for you from Chandrika Rao. She asks about the patterns you showed. That will the pattern of irregularity in the action potential help in the diagnosis of a particular neurological disorder?
Okay, so it may not help in the diagnosis, but as I already said, there are differences in a healthy brain and in a disorder called Fragile X syndrome, which is a autism level disorder syndrome. So as you saw, this is a recording from a healthy neuron,
Right.
and this is a recording from a Fragile X syndrome neuron. So as you can see, there are two differences I want to point out here. One is that there is a decrease in the healthy neuron is firing more action potential or spikes in the same time.
So the number of spikes and the duration.
And also if you see that 1, 2, 3. So 10th spike, say the 10th spike is occurring very, very close to each other in repeated trials.
Okay.
But in Fragile X syndrome, say 1, 2, 3. So 10 spikes are so much broadly distributed in different trials.
Okay.
So there are such differences that will help in our understanding of this disease and how to go about making drugs that will alleviate the symptoms.
Okay. Thank you so much for answering the question.
Yeah.
We hope, Chandrika, that your query was resolved. And thanks again, Anil and Anzal, for giving us a really good insight of your work today. And I hope everyone who was present with us today had a great time. And please do write in the comments if you would like to know or have any tour of any other specific lab at NCBS, or you would like us to do any other communication stuff for you. And we have other lab tours as well, which you can find on the Facebook page or Blisk India YouTube page. Thank you for joining us this afternoon. Take care, happy Holi, and very knowledge-filled Brain Awareness Week to you. Also, do check in NCBS_Bangalore Twitter page, because we are posting a lot of materials of what and how diverse neuroscience research is happening at our campus. Bye, take care, and have a great evening ahead.